This video we're going to start with the auger slants. Now there are all different kinds of tests, some broth, some are actually called gel plugs, and then some are the actual auger slants. And real quick introduction on how to work with like an auger slant and a broth, and then we'll get back to this. I'm going to start this next section with a video that shows, that looks like a broth tube. She is going to transfer something that's in this broth tube, and her aseptic technique is terrible. But basically right now, look, she is, now there's an auger slant. Now she basically just took some culture from an auger slant and she's putting it into a broth tube. And I wanted to show you this video because it was short and at least you can see what she's doing. If she were going to transfer something from in here to an auger slant, she would do the same thing, just straight from the bottom all the way up to the top. And there's some Kovacs reagent. Oddly enough, that's what we're going to be talking about here in a minute. But I just wanted to show you, when you inoculate tubes or anything like that, you would do a procedure kind of what she had done there. You would sterilize your loop, heat the barrel, take your loop, take some culture, and either streak a slant or you would swish it around in a broth tube. Now, once you inoculate auger slants, you're going to then put them into racks, and that's what you would keep them with on the table. Same thing, just a real quick reminder or introduction to what is a rack? What are you talking about with the racks and the test tubes, that kind of thing. I wanted to show you also, the name of this video was test tube holder and heating. But these are racks that you use in a micro lab. And yeah, there are times where you use the little tongs or forceps, they're called right here. And he's basically talking about heating the bottom of a test tube. We don't do that very much in our micro lab, but I wanted to show you these are the racks that when you're using different things, and we're talking about diagnostic media, if you inoculated 10 tubes, some SIM plugs, some broth tubes, you would then label your rack here with your name, your table number, the date, all kinds of things so that you can find your media. And then you would place these things in an incubator because an incubator speeds up how quickly they grow. You would do the same thing with the diagnostic plates. So any of these things we're talking about in diagnostic media, these do not occur immediately. You have to let the organisms grow so that they can ferment the sugars inside of the media, and then you get the color changes. And there's a Bunsen burner. I think he's probably going to line up here and heat some of those forceps, it looks like. There's a striker thing. I just wanted to show you that as an example. So all these tubes and everything and the plates, you would tape the plates together, but all the diagnostic media, you would put them inside of a rack like that, put them in the incubator. You would then come back the next class period, or 24 to 48 hours, you would take the rack out, and then you would read out your results. Okay, we are going to start on the middle section of our diagnostic media. You have what is called a citrate slant, and how you use this, you would take some culture, either from a broth tube, a plate, or an auger slant, and you would then streak the actual slant. You start at the bottom and you zigzag all the way to the top. You use what is called aseptic technique. Now, when you go to put these in the incubator, you would keep the caps pretty loose. They actually need some oxygen for this. And it says useful in distinguishing between gram-negative enteric bacteria. Gram-negatives, certain category. Enteric means they're from the gut. Gram-negative enterics are part of the mammalian digestive tract. And what you're testing for here is, can your organism use citrate as a source of carbon? All organisms, including humans, we have to get carbon from somewhere. We feed on other things for that. Plants can make their own carbon. They're called autotrophs. They're self-feeders. But bacteria, no different. They have to have a source of carbon. Why is this important? Well, only certain microbes can break down citrate and use that as a source of carbon. Once again, it's another test in your tool belt. You're trying to isolate and narrow down your search of what possible organism could you have. If you're trying to, in this case, identify something that's unknown, these are one of the tests you would use. So it's differential for citrate utilization, meaning the agar is going to change colors depending on whether or not it can break down citrate and use it as carbon. Blue agar is positive. Green agar is the original color, and I put it up here. The original citrate is green. So if you have a positive test, it would be blue. Here is a plate. Don't use those much. That'd be pretty cool, though. See the blue side and the green side? This is a positive. This is a negative. Same thing here. This is citrate positive. This is citrate negative. And like it says there, negative positive. So if you were asked on a quiz, is this organism positive for citrate? If it's blue, you say yes. If it's green, you say no. TSI slants. And I will put this in your title at the top. This stands for triple sugar iron agar. So it's got 3 sugars and some iron, and you can tell a lot of different things with it. These are sterile. They are agar slants. Now these are actually from a company with the label, but I know that, and you can make, look, you can make these Word documents bigger or smaller to get a better look at something if you want to. But see the slant portion? So what you would do is you would take your culture microbe, your unknown you're working with. You would then do your aseptic technique. You would sterilize the loop. You would heat the barrel of this. Well, you'd heat the barrel of your culture tube. You would get some bacteria culture on your loop, and then you would then open this up, heat the barrel, put your loop down here, and you would streak it all the way up. You're trying to get as much surface area as possible. Heat the barrels again, put the lid back on it. Heat the barrel of your culture as well at some point, and then you'd sterilize the loop. So these are TSI slants, and they start out a red color. This media is differential for glucose, sucrose, and lactose fermentation, all 3 sugars. Glucose is actually a monosaccharide. Sucrose, lactose are disaccharides. Now, some of these media, when bacteria break down sugar, they produce acid and gas, and hydrogen sulfide is a toxic gas. So this also tests for that. And hydrogen sulfide is a black substance. So positive for H2S, blackened agar. Negative, no blackening. Negative for fermentation, red slant, red butt. So you're talking about the bottom. The bottom's a portion of the tube. No change in agar color. Positive for glucose only fermentation, red slant, yellow butt. Positive for any of the 2 or 3, the entire slant, including top and bottom, turns yellow. So let's interpret these results. So what is this first thing you see here? Well, you see a red slant. They start out red. See all that black? That's hydrogen sulfide. So that is definitely a positive. This one here, red slant, yellow butt, means that it is positive for glucose only. Why is that important? Well, only certain things can break down glucose and not break down lactose and sucrose. Now, this one may look a little crazy. Here's a yellow slant, yellow butt, so that's definitely a positive. Do you see how this one is pushed all the way up? This one produced so much gas that the media, the actual agar, is being raised up into the top of the test tube. So this one was also positive for gas, but it's not hydrogen sulfide, which is that dark. And this over here would be a negative. Red slant, red butt, whatever you had. And you can kind of see, now look, what you do with these is you streak the slant and then you stab it down like this. See where the line is, where the agar, the loop went down. It even tells you to do that up here somewhere. It's a funny term. Yeah, stab the butt, streak the slant. And yeah, you can interpret a lot of things from that. Phenol red carbohydrate fermentation broth. Now, you can add all kinds of different sugars. You can get some that are maltose, phenol red, mannitol, phenol red broth, lactose broth, all kinds. They come in all different ones. They're all red, and they contain phenol, which changes colors and reacts into a change in pH. So if you have an organism that can break down whatever sugar you have in here, it's going to cause a change in the color. I left this here. This is supposed to be a tube. These have little things inside. They're called Durham tubes. So the top of this is closed and the bottom is open. Those are used to trap gas. Once again, microbiologists are obsessed with gas. And if you have an organism that can ferment lactose, and the definition is within 24 hours with acid and gas production, once again, it limits your search. It narrows down some more. Some organisms will turn this a different color but not produce any gas. So a plus is yellow broth. Negative is red because it starts out a red color. Positive for gas production, bubble in what is called the Durham tube. This is the Durham tube. Negative, there's no bubbles. Over here on the left, we have a negative test result. And look, you can even see in the tube, it's full of the broth. There's no bubbles, so that's a negative. This is a positive for carbohydrate fermentation, whatever the sugar was or carb that was in the tube. Once again, these come in a wide variety of sugars and things you can test for. It even says it there. Many different carbs can be used: lactose, sucrose, maltose, mannitol, all of those. This one, see the little gas bubble in there? This is a positive for lactose. It's also positive for gas production. If you don't see the gas bubble, it's a negative for gas production. One little side note with these phenol red, there are times where an organism can partially ferment something. It's called a weak positive. Right here, I need to put a little weak plus. It turns orange, and you can see there's the Durham tube inside. You can barely see it inside there, and there's no bubbles in it. But side note, weak positives for phenol red broths, they turn orange. Urea broth. Now with this one, make a note, you are testing for urease, which is an enzyme. And basically, it's an enzyme that breaks urea, which is the byproduct of protein metabolism, to ammonia. And ammonia, well, you know, if you've ever had an ammonia capsule popped under your nose, ammonia is a very strong scent. Urea hydrolysis test. The original color is that. It's called salmon. It's kind of an off orange. It almost looks like that. Off orange color. A positive would be fuchsia or bright pink. It's actually a really pretty color. It's differential for urease production. Urease is an enzyme. So now on this test, you're testing for a very specific enzyme. Does your organism produce urease? Why is that important? Well, only certain things can do that. So if you put an organism in this broth and you let it incubate for 24 hours and you come back and it's this pretty color here, that's a positive. These are slants. Really haven't used those much. It's mostly the broth tubes. This is a negative test result. See, that's salmon colored. This is a positive. What is it positive for? It's positive for urease, an enzyme. So this test right here, you're testing for an enzyme. You're testing for a very specific thing. Yeah, urease is an enzyme that splits urea to ammonia. And when it does that, you get a change in the pH, okay, the acid production or lack of, and it changes the color. One more, and then I'm going to go to the next video. So rabbit plasma, it's like it says. It's plasma from a rabbit. So what do you use this for? You use this to determine the difference between some Staphylococcus species, or if you're wanting to test to see if your organism produces something called coagulase. Coagulase is an enzyme that coagulates blood. So coagulase, yeah, causes blood plasma to clot. ASE means enzyme. So you're testing for the presence of coagulase in this particular test. Useful in distinguishing between Staph aureus and Staph aureus from other staphylococci. This is a liquid plasma. It's a clear liquid. A positive test causes this to clump together like jello. So differential for coagulase production. Positive, clotting turns the plasma to a thick gel solid state. Negative, plasma remains a liquid. Here's a great one. Look at that. See how that's, okay, here's the liquid. See how it's at an angle. It's actually a liquid, and then see how this is all stuck together because you hold it at an angle. Of course, liquid's going to flatten out. Something solid is going to stay in the end like that. So this is a positive, that's a negative. Staph aureus is positive for coagulase. See, staph epidermidis is not. See how it's at an angle? That's the fluid version. This is the thick, clotted version. Okay, I'm going to start there next.